Okay, hi everyone, and welcome to day 3 of the online microsensor workshop. My name is Line Daugaard, and I'm product manager here at Unisense. Today, Taya and I have moved to the lab, and we will do demonstrations of the microprofiling system and the microrespiration system. So I will just share my slides here. Yeah, so we are here on day 3. I will start with a demonstration of the microrespiration system, and then Tay will do a demonstration afterwards of the microprofiling system, followed by a presentation that shows you how you can analyze the profiles. So this is the program for today. Okay, so I will start and talk about the Unisense microrespiration system, and then I will do a live demonstration here on the table where I will show you the different parts of the microrespiration system. I will do a calibration of an uptone microrespiration sensor, and then we will have a look at a yeast solution, and we will measure some respiration rates. And afterwards, we have time for your questions. Okay, so the Unisense microrespiration system is used to measure respiration rates and metabolic rates in closed systems. Here you can see some examples of different sample types. We have bacterial cultures, cell cultures, small eggs. It could also be a small animal inside of the chamber. There are many different options. So with the system, you can measure respiration rate and metabolic rates in a closed chamber. All the Unisense sensors are compatible with the system, so you can measure all these different analytes. And you can measure with 1 or 2 analytes in one volume. And you also have the possibility to inject a substance into your sample and look at the immediate change of the metabolism. The system is designed to be submerged into a water bath to have temperature control. So the whole system should be completely covered by water, and then you have temperature control. Today, when I do the demonstration, I will work here on the table, but only so that you can see what I'm actually doing. In real life, you should make your measurements in a water bath. Okay, so I hope that you can all see the microrespiration system here. I will just again show you the sensors. They are all mounted in this blue aluminum guide that has a plastic cap at the end. And this cap ensures correct insertion into the lid of the chambers. When you have positioned the sensor in the lid of the chamber, you can then take out the sensor tip, and it will then measure inside of the chamber. When you then are going to move the sensor between samples, you can retract the sample tip and protect it. Okay, so we sell these different glass chambers. They go from 400 microliter and up to 400 milliliter. And I just have some of them standing here. We have many different types. For instance, we have this double chamber that allows you to measure with 2 sensors in one volume. And we also have these larger chambers. This is a 40 ml chamber, and that is the largest chamber that fits into the stirrer rack here. If you are measuring respiration rates or metabolic rates on a small egg or a small animal that needs to be protected from the magnet at the bottom, you can add a small metal net to the bottom of the chamber, and you can then put your sample on top of this metal net to protect the sample from the stirring. We also have these injection lids, and they have an extra capillary hole where you can, with a needle, inject the substance into your sample. The diffusion in and out of the chamber is slow that the chamber is closed. And this is because the capillary here where the sensor tip is inserted is so long and narrow that we can, the diffusion is so slow that we can eliminate it from our measurements. So for the rack here, we have different types of plates. And they fit with the different sizes of chambers. So if you, for instance, are working with the larger chambers, you just put this larger this plate with 2 large holes into the stirrer rack here. Okay, and for the stirring, we have these small glass magnets, glass-coated magnets that you put into the chambers. Never use Teflon magnets. They can simply take up the oxygen and mess up with your measurements. So always use glass-coated magnets. Yep, so I will just remove the chambers here. Yeah, so here you have the rack. It holds the microsensor and it also holds the chambers, and it can hold up to 8 of the small glass chambers. If you're working with the larger chambers, it can hold 2 of them. The sensors are connected to the Unisense amplifier, and you can use all the different Unisense amplifiers with these microsensors. The stirring of the rack here is controlled from this box over here. It is the stirrer controller. And over here, you control the speed of the magnet, and you can choose between different speeds, and you can choose between different modes. So you can have the same stirring in all your chambers, and you can also have different speeds in your chambers. So there are different options over there that you can control. Okay, yeah. So now I will start the software, and the software is called SensorTrace RAID. Yeah, here we go. I hope you can see the software now. I have just opened the software, and here you can see that it has detected the FX6 uniamp amplifier that I'm using, and it has also detected the optode that I have connected to the amplifier. It has detected a temperature sensor that I have connected, and then there's also a pressure sensor, and this pressure sensor is sitting inside of the amplifier, and it measures the atmospheric pressure. Then we are going to put in how many glass chambers that we are going to use. So you can just add the number of chambers that you would like to use here. I will just use one. And then you have to put in the volume of the chamber. And all the glass chambers are handmade, and therefore you have to find the exact volume of the chambers yourself, and this will just be done by weighing the chamber before and after adding water. So you have to find the exact volume, because the software will use this volume when it calculates the rate for you. Today I will use a 4 ml chamber, so I will put in 4,000, and this is in microliter here. Then you can give your chamber a name. I would just call it chamber 1. And if you were using a double chamber, you should then click here. I will just use a single chamber. So, okay, so then you press Start Experiment. And then you're asked to save the file. So I will just save it. Good. And now there's a warning saying that the possibility for data analysis is limited for sensors that do not use micromole per liter as unit. So in the beginning, you could choose different units. And if you want the software to calculate the rate for you, it's important that you choose the unit micromole per liter. So it's just a reminder about that. Okay, so the first thing we're going to do is that we are going to calibrate the optode. So I will start here in the calibration tab. I'm using a temperature sensor that I have connected to the amplifier, because then we will have an automatic temperature compensation. So I will choose the temperature sensor here. Salinity, I will just put 0. I'm measuring in water today. And then here you can see the pressure sensor sitting inside of the amplifier. So now I will press New Calibration, and it will then ask me to calibrate in an air-saturated solution. So now I will move the sensor to an air-saturated solution. Yep. And we have the calibration chamber here, where I have bubbled the water with air. I have inserted the temperature sensor, and now I'll just move the optode as well. down here, you can see the live signal, and I'll just click off the signal from the temperature and the pressure sensor. You can have a closer look at the signal from the optode here. Remember that the optode's raw signal is in degree because we're looking at a phase shift of the light. But when we have calibrated the sensor, we can get the unit micromole per liter. So now we wait for a stable signal, and we will then press calibrate air saturation. Good. Then I'm asked to calibrate 0. This means that we are going to calibrate in a solution without any oxygen in it. So I will just retract the sensor tip so it's in a safe position. Yeah, can leave the calibration chamber. Good. So over here I have a 0 calibration chamber, and this chamber has a special lid with a silicone tube. And I have filled the silicone tube with water, and then I have filled up the glass chamber with an ascorbate solution that has no oxygen in it. When I then put the lid with the water and the silicone tube into the ascorbate solution, as over here in the rack, the ascorbate will remove the oxygen from the water inside of the silicone tube. This means that we can now measure 0 point, so no oxygen, but we do it in water. So the water inside the silicone tube has no oxygen, and this allows us to directly afterwards move the sensor into our samples without having to rinse the sensor tip. So this is quite neat. So I will just put the sensor tip into the chamber here. So now you can see the signal changing here, and we will just wait for the signal to become stable. Yeah, and then I'll press calibrate 0. Okay, so now the calibration is shown down here. You can see the time point as well, and you can always see which calibration that is in use. You also have the possibility to push the calibration to the sensor. The sensors have this EEPROM memory, so you can actually save a calibration on the sensor memory. Okay, good. Now I will press calibrate it down here in the live window, and we will now see the signal from the sensor in micromole per liter. Good. So now the sensor is calibrated, and we can move on, and we can measure our yeast solution. So I'm going here to chamber 1. And here you get the possibility to choose how often you would like to have a data point. So by default here, it's set to 1 data point per second. But if you know that you have a very slow respiration rate and you would like to measure over a longer time period, you can slow this interval down. When you're using optodes, this can also be quite relevant because then you will save the dye. There's a certain number of data points coming with an optode. Okay, but I will go to the camera again, and then I'll prepare a yeast solution that we can measure. I have a yeast solution prepared here. Good. So I will take a glass chamber here, and I have added the magnet to the bottom of the chamber. Then I will take the yeast solution and pour it into the chamber here. And it's important that you fill it up completely and that you keep an eye on air bubbles. You don't want air bubbles inside of your sample. So I will just remove a small bubble that I have here. Yeah. And then you add the lid, and it will spill over, but this makes sure that you have liquid all the way up through the capillary hole in the chamber. So it has to spill over a bit. Always have a look at your sample and check that you have no bubbles inside of your sample. You don't want to have an air phase in your sample. Okay, so now I add it here to the rack. Because I'm doing it on the table today, I will also add just a solution of water that I can put my temperature sensor in. If you're working in a water bath, when you're doing that, you can just add the temperature sensor into the water bath close to the rack. Okay, then I will move the sensor. Yeah, and then we can start measuring. Yeah, so you can always stop your measurement and start it again. If, for instance, if you're moving the sensor to a different chamber, you would then stop the measurement, go to the next chamber and start the measurement, and then you could go back again. So you can move the sensor between your samples. And now it will just ask me if I want to use the same sensor. Yes. You can always zoom in on the data and you can zoom out again. It will all the time try to make a regression line here. I think I might have just one off point here, but so you can see that regression line is a bit off, but it is doing a live regression line here. When you stop the measurement, you can do a manual calculation where you move the slide bars here. So if you, for instance, want to have a closer look at an area here, you can then over here see the slope and the standard deviation. So you can just move the bars and have a closer look on your data here. Yeah, just start it again. Yeah, but I think you can see that the oxygen is slowly decreasing in the chamber. So the yeast are eating some oxygen. In the tab here called rates within, you can see it calculates the consumption and the production rate. And it will give you these blocks, green being production and red consumption. Down here, you get a table that gives you the rate in nanomole per liter. And you can see the different blocks here, M1, M2, M3. These are the 3 blocks that you can see up here. You can then add the slope and the 95% confidence level, the average concentration, and the start and end point as well. So then you get the data over here. You can export it to Excel or as a CSV file. So then you get the data. Yeah. You can also go to rates between. So this could be relevant if you move the sensor to a different chamber and you're going back again, and then you can have a look at what was going on during this time. Down here, you can choose how many samples before and after the measurement that you would like to include when it makes this regression line here. So you can play around with that as well. The same here as for rates within. You can add the different slope, 95% confidence level, and the mid-time concentration, and start and end points. And again, you can export this table to Excel and a CSV file. The last tab here plots the rate versus concentration. And over here, you can see comments, so what was going on. You can always make a comment during the measurement. I think I'll just go back to the chamber here, start measuring. Then you can always make a comment. So I would just call it yeast respiration. and then it will add it with the current time. You can also choose a different time point here, but if I click here, it will use the current time point. And then when you go to comment, it will then be seen here. So if you change the conditions or you add something to the sample or whatever, then you can make a comment so that you can remember what was going on in the chamber. Yeah. Okay. Yeah, so you can see in the live window that the oxygen is decreasing, so the yeasts are using up the oxygen inside of the chamber. And because the chamber is completely closed, at some point the yeast cells will have used up all the oxygen inside of the chamber. Okay. Yeah. So now I will go back to my presentation. Yeah, it's just to show you the different sizes of glass chambers that we have. In the MicroRespiration System manual, you can find this overview here where you can see the volumes and the diameters of the different glass chambers. So you can always go here and have a look and see what chamber that fits with your sample. Here you can see a picture of the double chamber that I showed you where you can insert 2 sensors and measure in one volume. You can actually also measure in this triple lid that is seen over here to the right. It has 3 lids at the top here, and it's possible to measure with 3 sensors in this chamber. Then I would also just like to show you this slide here where we have the uptode and the Clark type oxygen sensor, a comparison of those two. For microrespiration system measurements, we very often recommend uptodes for respiration rate measurements. The uptode requires less frequent calibration compared to the Clark type. So this can be convenient when you're looking at respiration rates that are slow and you have some long-term experiments. The uptode also has no hydrogen sulfide interference and it doesn't have any noise interference or handling noise. So this can be quite convenient. On the other hand, the Clark type oxygen sensor has a wider concentration range and also a wider temperature range and a lifetime that is not limited by the dye at the tip of the uptode. So there are pros and cons, but typically an uptode can be a good choice if you're going to measure respiration rates. Yeah. So now I will ask if you have any questions. You can put them in the chat. You're also very welcome to unmute yourself and ask your question. There's a question about how we make the escapade solution. There's actually a protocol for that in the manual for the oxygen sensor. So you can go and have a look. It's on our website. We also sell a calibration solution with a zero solution. So that's also an option. Then you get 10 vials with this zero solution. But of course, you can prepare it yourself as well.
And I actually think there was a sort of an easy way to do it way back. So a teaspoonful of sodium ascorbate and 1 pellet of sodium hydroxide into 50 milliliter of water and shake it. And then, so that's easy.
Then you have it, yeah. Switch, okay, yeah, so switch from between 2 different vials, yeah. I will just share my screen. Yeah, so if you have added 2 chambers in the beginning, you would have a second tab up here saying chamber 2. So I would then stop the measurement in chamber 1. Then I would go to the tab with my chamber 2 and then start the measurement. If I then wanted to go back again, I would then stop measuring and then go to the chamber 1 tab and start measuring again. And then it keeps track of the measurements in the different chambers that you have.
Yeah, so there's another question about moving the sensors between different chambers. And well, there's no handling noise in the uptodes. But there is for all the amperometric sensors, so the oxygen, hydrogen, sulfide, and so on, because the the signal in the wire is so low, it's in the picoamp range, and just bending or moving a cable will generate a little bit of signal. So that's what you see when you're moving it. But it doesn't affect the sensor or anything, and just as long as you just keep the cables stable while you are measuring, then it's not a problem at all.
Yeah, so you don't see it on the actual measurement, because when you have positioned the sensor in your vial, it's completely stable, and then you press start measuring in the software. So it will only show you the noise when you move the sensor around. Yeah, so there's a question about how often should we calibrate the sensor, and that's a really good question. We always recommend that you calibrate the sensor before starting a new experiment. The signal will change a bit over time. So if you're measuring for a longer time period, it can be a good idea to do the calibration again after your experiment, because then you would have an idea about how much the signal has drifted during your measurement. But we always recommend to do a calibration before starting a new experiment.
And there's a question about the volume of the chamber which you put in. Line, you put in 4,000 microliter, and
yeah, that is the volume of the glass chamber that you're measuring in. You don't have to put in the volume of the calibration chamber. It's only the glass chamber where you have the sample, and that's because the software uses this volume to calculate the rate in nanomole per hour.
And about cleaning the sensor, how do we do that?
Yeah, so the tip of the sensor you can clean with ethanol or any detergent. The glass chambers, they can be cleaned with soap and water, and you can also autoclave them. And the rack, you should clean with soap and water as well. You cannot autoclave that, but the glass chambers you can autoclave, and the sensor tip you can rinse with different solutions. Yeah, you can also use acid as well. Yeah, different proteinases, and yeah. There's a question if you can use the double chambers with a single sensor, or do you need to use two sensors? So you add a lid to both sides of the double chamber, so you have two lids on top, and you can measure with one sensor, or you can measure with two sensors. You don't have to put in two sensors. So thank you for now, and remember to leave this meeting and enter the new meeting link.
That's one more question.
One more question,
Yeah,
yes. We have time, so
yeah.
Yeah, what would we recommend for volumes of 400 milliliter? Yeah, so it's true. The largest volume that fits with the stirrer rack here is 40 mils. So if you're going to use larger volumes, you would have to find okay. But for larger volumes, you would have to find your own rack and holder for the sensors. For the 40 mil, it fits into the rack, so you can have two 40 mil chambers in the rack at the same time.
And it doesn't need to be anything fancy. A standard magnetic steer plate will work well for steering, and you can just have a standard lab stand with a clamp on you that can hold the sensor in place. So it's just not so difficult to do it.
There's a question if you can use a needle sensor. No, you have to use the microsensors here that are mounted in this blue aluminum guide with the plastic cap, so that the sensor is inserted correct into the small capillary hole of the lid of the chambers.
Yeah, the smallest needle we have is 1.1 millimeter in diameter, and the hole in the lid is 0.7 millimeters.
0.7,
so that's a problem.
Yeah, it's very narrow. Thank you very much for attending.